Thank you. It's great to be here to be able to tell this story. This is kind of a newish story from my group that's kind of brings in new things, and so it's great to be able to tell it to this crowd. It's a work in progress, so if people have comments or new ideas, new directions, I'd love to get that feedback. Okay, so this is a collaboration with Ben Macda's group, who's someone I've collaborated with for a long time. He's a professor at Yale, and this was the thesis project, or one of the thesis parts of the thesis project of Mason, who is now a postdoc at the University of Chicago. And the experimental work was done by Yusuf, who is a postdoc in my group. Okay, hopefully this works. Okay, so if you've been paying attention to the cell biology, maybe for the last 10, 15 years, it's been all aflutter with concepts of phase separation being involved in all sorts of biological processes. And this is for good reason. It's really cool. It's really kind of attractive to think that this physical concept of phase separation could be involved in organizing all sorts of kind of functional processes within cells. And the microscopy evidence for such things, it's just so striking and so beautiful. These phase transitions that happen in cells is driven by kind of standard polymer physics ideas. These are biopolymers that condense because there's multivalent interactions between them. And it kind of follows all the rules that you would expect kind of from a polymer physics perspective. So the talk today is about thinking about how these condensates, these biopolymer drops interact with surfaces. And here's a picture of actually some of these protein drops interacting with a cover slip slide. And if you just look at it, you would think it's just water drops on a windshield, but in fact, it's actually a super complicated mix of proteins that are involved in the postsynaptic density of neurons, but sitting on a cover slip kind of bathed in water. And it looks just like you would expect other types of drops wetting a surface. These polymer drops can wet other types of surfaces too, and membranes is one of those surfaces that I care deeply about. And I think I saw that not too long ago, Rumi Damova, who's kind of done extensive work in this area, already gave a talk in this series. But she's done a beautiful work looking at wetting of these biopolymer drops onto membranes and the various behaviors you can see there. And so one of the things you see is that these drops can kind of deform membranes, and the membranes can deform the drops. But my talk today is about something called prewetting. And I will explain what this means in a minute, but I just wanted to give you a bunch of examples of it from the literature. These are all examples of proteins that are involved in condensates, where one part of it has been stuck to a membrane or forced to interact with a membrane. And then when this happens and you can drive this system to condense, you get beautiful phase separation that's driven by these protein interactions or biopolymer interactions. But now it's happening at the membrane surface, and it's effectively a two-dimensional thing. So it happens for all sorts of different systems. So I'm giving you many examples. This is proteins involved in immune receptor clustering, the signaling underneath immune receptor clusters. These are postsynaptic density proteins. This is actin-related proteins. These are growth factor signaling related proteins. This is a protein that people love to study in condensates, and they've just stuck to a membrane because they can. But there's many examples of this all over biophysics, and it's really striking and beautiful. Okay, so I said I'd explain the difference between wetting and pre-wetting, and so here it is. So wetting happens when you have a bulk condensate, so a polymer drop in solution, and it interacts with a surface. So in this case, it's interacting with a membrane. It happens above this CSAT of the drop, so it's stable in solution. And it's a 3D object, and it extends into the bulk. Pre-wetting, on the other hand, happens below CSAT, so it happens under conditions where the condensate is unstable in the bulk. It wouldn't form a drop on its own, but it forms this condensate at the membrane because the membrane is there. And because it's not stable in the bulk, it doesn't extend into the bulk, so it's effectively a two-dimensional structure. It's a surface phase, a surface phenomena. Okay, so why does this happen? there's just a bunch of kind of very intuitive reasons. One is that by often tethering one of these components in the condensate to the membrane, you just increase its local concentration. And so, effectively, it's higher concentration, so it doesn't take as much to cluster it. The other kind of more subtle thing is that by restricting one of the components maybe to the membrane, you've restricted its conformational entropy. And so clustering it is less of an entropic cost. And so it's also more likely to happen. Okay. So I've shown you many beautiful examples of real proteins condensing on membranes. We can also do this with very simple polymers. So this is polylysine and polyglutamic acid in cartoon form, but we can also do it in a real experiment. And so this is a multi-layer of a boring membrane, a DOPC membrane, where we have a small fraction of lipids that are clicked using click chemistry to polylysine. And then we add polyglutamic acid and polylysine to solution, and we can get pre-wet domains to form. This is a multi-layer mess of an experiment, so just bear with me. It's a little messy of an image, but I thought I'd show you the real experiment. So you get multi-layers, so you get different shades of gray of our probe. You also get a lot of defects in these particular multi-layers. But the main point is that we get these pre-wet domains that kind of can easily diffuse around on this surface and do all the things we want domains, phase-separated domains to do. So they can diffuse and they can merge eventually, will happen in this movie. This system is nice being so simple that we can model it also and do simulations to kind of recapture some of the behaviors that we see. Okay, so that's the backdrop. And so now we have this very simple system. We have lots of knobs that we can turn, and we can map phase diagrams. And so that's what this is. And so we varied polymer concentration in solution, and then we've also varied salt concentration, which kind of titrates the interaction energy between these polyelectrolytes. And we can be in a regime where there's pre-wetting, where we get these domains at the membrane. And the one reason we know they're pre-wet is that we don't see them in a transmitted light image because they're not big enough to diffract light on their own. But if we increase the concentration of polymers in solution, we can drive it towards wetting, where we start to have three-dimensional drops in solution, and then we start to see these drops by transmitted light. Okay, but the important thing for this talk is that this pre-wetting is happening, in this case, about at 20-fold less soluble polymer in solution than where we see wetting, which is where you get the bulk of phase separation. So this is really robust, happens well below the 3D bulk phase transition. Okay. Membranes can also have a phase transition of their own, and it's driven only by lipids. So this is a model membrane that has 3 different lipids in it, and we can lower temperature in this case, and it can break out into spots. These spots are different phases, just like our other system with biopolymers, but now there's no biopolymers involved. It's only lipids. And it's in the plane of the membrane, so it's effectively 2-dimensional. Yes, and these phases have different names, but it doesn't really matter at all for the purposes of this story. One interesting thing about phase separation in membranes in 2 dimensions is that near the transition, you can have a very high susceptibility. And just bear with me, this high susceptibility comes across in a bunch of ways, but one way that's really easy to see it is in the context of fluctuations that happen near a critical point. So when the susceptibility is high, one of the consequences of that is that the free energy of forming a domain becomes small. And when it becomes small enough that it drops below the thermal energy, it actually happens spontaneously. And when you're very close to the critical point, the domains that can form spontaneously become very, very large. They can be 100,000 times bigger than an individual lipid. And that's why we can see them in a regular old microscope image. And what's changing in this video is the temperature, which is changing the magnitude of the susceptibility. And that's why we see the change in the size of these fluctuations. When you're further away from the critical point, the susceptibility is still big, but it doesn't give you this gigantic of fluctuations, but they can still do useful things. And the free energy cost to pre-wetting, right, to clustering things on the membrane is related to this susceptibility. And so maybe this is going to matter for this particular measurement that we're trying to make. Okay. So this was the kind of a revelation that Ben and Mason had at the beginning of this project. And so they sharpened their pencils and asked the question, how does being near this membrane phase transition affect when pre-wetting happens? And so they did a bunch of calculations and a bunch of simulations and indeed convinced themselves that when you're near this membrane phase transition, you should just dramatically enhance the regime where you see pre-wetting. And they did this with mean field theory, and they also did this with simulations. And this movie is not my favorite because it's not super clear, but what's changing in this movie is the membrane kind of parameters. And at some point you can see there's a domain that forms of these biopolymers on the membrane. I have some better pictures that will come. So they showed this in theory, and then we wanted to try to do this in experiment as well. So we just took the same system that I already explained to you, but we added a few more lipids in. And we chose compositions that were near the phase transition, but above it, but different distances from the phase transition, both in composition and temperature. So this is our membrane with no phase transition, and we get pre-wetting happening at one particular concentration of soluble polymers. But then as we walk closer to our phase transition, we notice that you started needing fewer and fewer polymers in solution to start seeing these pre-wet domains. And if you map out the whole phase diagram and vary it with salt too, you can kind of see this is a really robust effect. You need a lot less soluble polymer in solution when the membrane is very close to its phase transition. We can gain a whole nother order of magnitude in polymer concentration. And you can see this kind of acutely if you start off with a membrane that has low cholesterol concentration in this case, and we acutely add cholesterol to the membrane, we can go from a system that has polymer drops on it, and simply by changing the membrane lipid composition, we can melt those drops away. These pre-wet phases are real phases, and they incorporate both these polymers and lipids. They have a different lipid content and a different polymer content. And if you lower temperature, so this is above the membrane phase transition temperature, if you go below the membrane phase transition temperature, you can start seeing three-phase coexistence. And these are also three phases of all the things that are in this system. So it's a really complicated system, so you can have many phases. A few extra tidbits for maybe people that are interested in details. So if we start off again with our boring membrane that just has one component plus our tethers, and then we switch out that liquid fluid membrane for one that forms a gel phase at room temperature now, or at 37 degrees. Now we lose pre-wetting altogether. So you need a fluid membrane in order to have this pre-wetting. We can walk towards the phase transition, but not go as far as I did in the other pictures, and still see that that pre-wetting is potentiated. And this is just to say you don't need to be anywhere near this phase transition for this to matter. You really don't need to be near a critical point either. It's just a really robust effect. And then one other interesting point is that here we just walked from DOPC, we added cholesterol to the membrane, and we actually saw that it got inhibited, which I don't fully understand this. This is a kind of a confusing result to me. So if people have ideas, I would love to talk about it. Okay, so where we stand so far, I've told you that there's this phenomenon called pre-wetting that happens at membranes. It happens below the CSAT, so it happens when the polymer is not forming drops in solution, but it forms this condensed phase only on the surface of the membrane. This is really a really robust kind of surface phase that happens in this context of where you have this biopolymer phase transition that interacts with a fluid membrane in particular, and there's lots of beautiful examples in the literature. Our contribution is to look at membranes that also have their own phase transition, and we found that when the membrane has its own phase transition, it can dramatically enhance this pre-wetting even more. And the important thing that I want you to take home is that this effect happens even though both of these systems are very far from their own phase transitions, but by coupling them together, we get a new phase transition that's a real phase transition. It does its own stuff. Okay. So we were interested in thinking about how this kind of connects to cell biology. So we brought this system into cells, and we used an existing system that people have published on before. It's kind of complicated, and don't worry about the details, but we have 3 different things that we express in cells. We have a optogenetic bit called Cry2. It sticks to a GFP that we can put wherever we want. It has an anti-GFP nanobody. So those are 2 things that we express. And then the third thing we express is called CYB1MP. And it's basically a very multivalent protein that interacts with Cry2 when it's photoactivated. Okay, so in this system, when we express all the things together and we expose it to light, the cells will break out into spots. These spots happen at the plasma membrane when we co-express with a plasma membrane-bound GFP. And these are beautiful phase-separated, fluid-looking protein domains. They diffuse around, they coarsen, they do all the beautiful things that, as a membrane person, I always wanted lipids to do long ago, but they never did. But this system is really well behaved. Okay, so we have a system. We wanted to come up with an assay. And so what we do is we co-express these 3 constructs. They all have different fluorophores on them in U2OS cells, a cell line that's very amenable to transfection. And we end up with, it's transient transfection. So all these cells are expressing different levels of all these things. So it's a big mess. But we transfect a whole bunch of cells together. We split them into lots of different dishes. And then we expose those dishes to different amounts of light. Chemically fix them, and then bring them to the microscope, image lots of parts of the field of cells, and then go back and score them for how clustered they are, basically. And so the number that we pull out at the end of the day is the fraction of cells that contain clusters in a dish. Okay, so when we do that, and we have our membrane-bound GFP, we see that we need some amount of light in order to see puncta in our cells. But when we express a soluble GFP, so now no longer attached to a membrane, now we need a lot more light to see these puncta. And it's about an order of magnitude more light. And this is kind of reminiscent of what we saw in our simple system of polyelectrolytes, right? We needed about an order of magnitude more soluble polymer in order to be able to see wet domains versus pre-wet domains. So that's why we think these are probably pre-wet. Another reason we think these are pre-wet is because we can do kind of fancy confocal imaging of our cells, and they look like they're just at the plasma membrane. And so this is just imaging the bottom surface and different confocal slices as we go up, and they seem to be only at the bottom plasma membrane in this case. But if we expose our cells to a lot of light, then we take confocal slices, and we can see these domains extend into the cytoplasm. Okay, so we're interested in this because the plasma membrane also has a phase transition. And we know this for lots of reasons. One reason is that you can do terrible things to cells and coax them to phase separate, their plasma membrane to phase separate. So you can bleb them and you can get them to phase separate. You can add detergent and get them to phase separate. There are other things you can do. You can extract lipids and get them to phase separate. But kind of on their own, they don't just phase separate. Another more subtle experiment that we've done in the past is to find evidence for the membrane being highly susceptible. And we did this with super resolution imaging recently. And I'm happy to talk about this more if people are interested, but there's evidence that it's kind of not too far from a phase transition based on these experiments. And there's also a lot of evidence out in the literature that it's not too far from a phase transition because there's lots of evidence for nanoscale heterogeneity, which I would call maybe fluctuations, evidence for it being easy to assemble small scale structures. Okay, so we're wondering if maybe this membrane phase transition in cells impacts pre-wetting. Okay, so the first thing we did was change temperature. It's like the easiest thing to do. So we took our cells and we exposed them to light at different temperatures and chemically fixed them and did exactly the same assay and found that when we lowered the temperature of our cells, less light was needed to form puncta. And when we raised the temperature of our cells, we needed more light to form puncta. As a control, of course, we want to look at soluble GFP too, so something not tethered to the membrane, get the membrane out of it. And not surprisingly, we do see some effective temperature on that system as well, because temperature is important for all thermodynamics and not just membrane thermodynamics. And so it's a little bit complicated to interpret this as just a membrane-based phenomena. But it's still fun. You can do the experiment. So here's a live cell now. We've taken, it's starting off at high temperature. We shine it with a little bit of light. but not enough to form puncta. And then we quench the temperature down, and you'll see that puncta form. Get a drink. And our stage doesn't deal, our autofocus doesn't deal very well with temperature changes, so the focus keeps going in and out. But there's puncta there. And then we raise temperature again, and they go away. There. So reversibly, we can get these drops to form. Okay, so temperature is not ideal. It affects lots of things, not just the membrane. And so we moved to another type of perturbation that we've used a lot in the past. We use these N-alcohols that we think perturb the phase transition temperature, but don't affect other things as much. And so one that we like is called hexadecanol. It raises the phase transition temperature of the membrane, whereas octanol, which is a shorter chain N-alcohol, it lowers the phase transition temperature of the membrane. And we think for a bunch of reasons that it doesn't do a lot of other things. It changes the transition temperature, but doesn't change the membrane in other important ways. And so we just chose one combination that we've used before. Okay, so when we add hexadecanol to the membrane before we do light activation and fixation, we find we need less light is needed. And that brings it closer to the phase transition. And when we do octanol, we need more light to start seeing puncta. And this octanol brings it further from the phase transition. But in this case, when we do this soluble GFP control, we don't see any effect. And these are hydrophobic molecules. They're probably just stuck in the membrane. We're not expecting them even to touch these soluble condensates. So here's an example of a live cell that's been we pre-illuminated it with light, so you get puncta, and then treated it with octanol in this case, which lowers the membrane phase transition temperature, brings you further away from the membrane transition, and it's going to melt these domains. Fun. And what was really satisfying for me is that when you look at the temperature, real temperature results and these N-alcohol results right next to each other, they qualitatively look very, very similar. So it kind of supports the idea that these N-alcohol perturbations really are having an effective change of the membrane transition temperature without changing real temperature. Okay, we can do other things to perturb the membrane. We can change cholesterol concentration in this case. That also affects the amount of light that you need in fixed cells, and then we can also do it in live cells. And so when we add cholesterol to live cells, we can trigger this phase transition. And now we're looking actually at the top of these cells, so you can see these domains move around a lot more. It's pretty fun. And then here's an example where we've added an ionophore that lets calcium into the cells. Letting calcium into cells does all sorts of things. One of the things it does is it activates a scramblase that lets relieve some of the transmembrane asymmetry. It also does a lot of changes PIP homeostasis. But in this case, when we add this inhibitor, this ionophore, we also melt these domains. So there you go. Another example, so there's nothing that forces us to stick with the plasma membrane. We can put GFP on the ER in this case, and we can do the same tricks. And so we did the same kind of assay, but now we're sticking our condensates to the ER. And we find that we need kind of an intermediate amount of light in order to be able to see puncta in these cells. And we don't know why. It could just be that we're expressing different levels of this protein. I'm not sure. It could be that the membrane is fundamentally different. It could be something more interesting. I don't know exactly. So this is something we're interested in. But one thing we've noticed is that if we add cholesterol to these ER tagged cells, we can also trigger pre-wetting of the ER membrane in this case very quickly. And so I'm not sure what's going on here, but it's kind of fun. And so I don't know if increasing cholesterol is leading to the ER membrane looking more like the plasma membrane, so now it needs less light, or if maybe we're inducing some stress response that's triggering this, or maybe something else entirely is going on. I'm not sure, but something interesting. And then the last thing, and I'm almost done, I promise, is we can now put our tags on both the ER and the plasma membrane, and do the same experiment basically. And then when we do that, we kind of just use the system. It ends up being glue to glue these two membranes together, and we make contact sites. And so we bring in the ER in close proximity to the plasma membrane through this condensate glue. It happens at intermediate light concentrations to both amounts to both the plasma membrane and to the ER. And it's also sensitive to this perturbation of adding octanol in this case. So it seems to care about the lipids, not just the proteins. And I'll skip this. Okay, so where things stand. So we have this kind of fun new tool that we're kind of learning how to play with, where we can titrate this pre-wetting interactions with light. We can play with it in ways that very similar to our very simple models. And we are kind of seeing things that look a lot like our model system. We see what looks like pre-wetting, and we also see the membrane sensitivity in ways that are very similar to our model system. We can do this at the plasma membrane, but we can also do this at other membranes, and we've done this with the ER, and we're also playing around with ER plasma membrane contact sites. And the important thing, I think this is the real big important take-home message, is that this real phase transition happens even though both of these systems are really far from their individual phase transitions, right? The plasma membrane is not undergoing any phase transition anywhere close. These condensates aren't undergoing their phase transition anywhere close. Yet together, we see this really interesting structure. Okay, so all of this is to say that by coupling these two things, the membrane gives you kind of this high susceptibility of membranes, makes it easier to organize things, whereas this condensates give you all of these specific interactions that help you cluster things. And by coupling these two things together, we get this completely new phase transition that you could imagine driving all sorts of biological processes. And I think we've just like scratched the surface of this. I think it's a really fun, exciting new area. And so with that, I would like to thank you for your attention. Thank Ben for really driving this forward. This is an old picture, but it's got everyone in it. So Ben, Mason, and Yusuf. Thanks.